cell and how epinephrine affects it. So we have epinephrine, which is the hormone messenger that is given off by the adrenal glands. Epinephrine is then going to go and dock with the G protein receptor. The epinephrine is known as a ligand, which is a chemical that cannot make it through the phospholipid bilayer. The G protein is a family of proteins involved in transmitting signals from stimuli outside of the cell to inside the cell. If we were able to see the real thing, the G protein would look sort of snaky. When epinephrine binds with the G protein receptor, it makes a conformational change, also known as a change in shape. When this change in shape occurs, it releases an alpha subunit. This subunit goes down the membrane to a protein called adenylyl cyclase. This protein is what changes the alpha subunit from inactive to active. Now that the alpha subunit is active, it can do its job as an enzyme. In this case, the alpha subunit converts ATP into cyclic AMP, which is known as a secondary messenger. ATP does this by dropping off two phosphates. These secondary messengers then target protein kinase. This protein has two catalytic portions which speed up chemical reactions, and then the regulatory portion. The cyclic AMP binds with the regulatory parts and releases the catalytic portions. These catalytic portions now pick up energy from ATP and are now activated. Catalytic portions are now going to act on enzymes in the cell. In this case, it targets phosphorylase. The dropping off of the phosphate activates phosphorylase and will now release glucose from glycogen. This glucose will be sent outside of the cell. Once the ligand is not attached, the process will be shut down. We are going to go over a quick recap of what's going on. So we have epinephrine, which is an enzyme. It's also called a ligand. It's coming down and it's binding with the G protein right here, which is also this blue little part right here. So we have the subunits that are also attached to the G protein, and when the ligand binds, it's going to then send a subunit all the way down to adenylyl cyclase. Now, once this alpha subunit binds with adenylyl cyclase, it becomes active. When it's active, it produces ATP. These ATP are then going to go and create cyclic AMP. Cyclic AMP is also a secondary messenger. This cyclic AMP comes over here and binds with the regulatory parts of protein kinase. This cyclic AMP then pulls the catalytic portion and the regulatory portion apart, which creates the catalytic portion being able to come down and combine with phosphorylase. This combining of phosphorylase then kicks glycogen out of the cell, producing glucose.